Okay, wait, we're good. All right, I'll be back in a second. Okay, but now the day is free and I have hours and hours to do homework. And I'm so grateful for that. Genuinely. Free time. What the fuck? They're so stale. Oh, okay. Transcription of the operand genes is usually on. When they say usually, no, wait. Oh, I don't know about this catabolic thing. Okay. Yeah, anabolic. You want to make it until you have the right amount. So anabolic is going to be repressible. And catabolic is going to be inducible. That makes sense. It's so stale.
E. coli cells live in our
This is
intestines
14 minutes
and
long,
use
bro.
glucose as their preferred energy source. When a person drinks milk, these cells encounter an alternative energy source, the sugar lactose. Although E. coli can metabolize lactose, it will make the proteins for lactose metabolism only when two conditions are met. Glucose levels are low and lactose is present. This adaptation saves energy and confers a selective advantage in that only necessary proteins are produced at any given time. An operon is a group of closely linked bacterial genes and regulatory sequences that produce a single messenger RNA molecule in transcription. To understand how E. coli regulates lactose metabolism, let's examine a cluster called the lac operon. There are three structural genes in the operon. The
Bro,
lac-Y gene
operon,
encodes the protein
you're gonna bother me.
galactoside permes, which transports lactose into the cell. The lac-Z gene encodes the enzyme beta-galactosidase, which cleaves lactose into glucose and galactose. Researchers are uncertain whether the other structural gene, lac-A, plays a role in lactose metabolism. The lac operon is regulated in part by a gene called lac-I, which encodes a repressor protein. The lac-I gene is always expressed if there is no lactose present in the cell. The repressor binds to a regulatory region of the operon called the operator. When RNA polymerase binds to the promoter, the polymerase cannot pass through the operator site to transcribe the structural genes. The lac operon is therefore under negative control, meaning that a repressor blocks transcription.
Bro, it's the same shit.
Lactose is considered an inducer of the lac operon because its presence can induce the expression of the structural genes. The inducer is really a derivative of lactose called allolactose, but for simplicity, lactose is referred to here as the inducer. Lactose works by binding to the repressor and causing it to change shape. In its new conformation, the repressor is inactive and cannot bind to the operator. This is an example of allosteric regulation. Lactose can also cause the change in shape while the repressor is bound to DNA. In this case, lactose releases the repressor from repressing the operon. The RNA polymerase can then transcribe the structural genes. The current understanding of the lac operon is based in part on experiments performed nearly 50 years ago by Francois, Jacob, and Jacques Monod.
I mean,
Let's
that's the thing
examine
about
some of
these
these
homeworks,
experiments
bro.
and the conclusion.
It's just like the same question over and over and over again. I mean, like, we just did what is laxy? We just answered that question. Why do I need to do it again? Okay, that I don't know.
Conclusions the investigators drew. To study the genetics of lactose metabolism, Jacob and Monod generated several classes of mutants. They exposed E. coli cells to X-rays, ultraviolet light, or chemical mutagens, which mutated E. coli's DNA at random positions on the chromosome. Next, the investigators spread the bacteria onto master plates to propagate the cells. The medium on these plates was complete. It contained a variety of compounds that would allow any of the mutants to grow. Each cell divided repeatedly to form a visible colony of bacteria on the plate. All the cells in a colony were genetically identical. To find mutant cells with defects in lactose metabolism, the investigators performed replica plating. In this procedure, a sterile velvet stamp is pressed onto the master plate to pick up bacteria from the colonies. The replica plate has medium containing only the sugar lactose as a carbon and energy source. The stamp is pressed onto the replica plate to transfer the colonies from the master plate. A control is also generated by stamping the colonies onto another plate with complete medium. Look at the pattern of colonies on both plates. Keep in mind that the investigators had to examine many more colonies than are shown here. Some colonies on the master plate contain bacteria with defects in lactose metabolism. Mutants in lactose metabolism are able to grow on complete medium, but not on a medium that contains only lactose as an energy source. The regions that could be mutated to produce cells that cannot use lactose as a source of energy are lac-I, promoter, lac-Z, and lac-Y. If the mutation resulted in a repressor molecule that is unable to bind to lactose, then the repressor would always block transcription of the lac structural genes, and the cell would not be able to use lactose as an energy source. However, another type of mutation could result in an opposite phenotype. If the mutation resulted in a repressor that is unable to bind the operator, the structural genes would always be expressed. If the promoter is mutated, such that RNA polymerase cannot bind, then the structural genes for lactose metabolism will not be expressed. Without these enzymes, lactose can neither enter the cell nor be broken down if it were to get in. If a cell has mutation in the lac-Z gene, it would not be able to produce a beta-galactosidase enzyme. Beta-galactosidase breaks down lactose into simple sugars, glucose further by other enzymes in the lac-Y enzyme. Now let's look at another way to understand mutants. To test for the normal function of the lac-Z gene product, Jacob and Monod used an indicator molecule called O-nitrophenyl beta-D-galactoside, abbreviated ONPG. ONPG has a structure very similar to lactose and can be cleaved by beta-galactosidase. But when this molecule is cleaved, galactose and a bright yellow compound, O-nitrophenyl, are released. The researchers now had a method of finding and analyzing distinct classes of lactose mutants. The ONPG technique allowed them to determine if the genes enabling lactose to be used were transcribed and translated even if lactose was not present in the medium. Observe colonies grown on a medium containing a carbon.
Wait, why?
Technique allowed them to determine if the genes enabling lactose are released. The researchers now had a method of finding and analyzing distinct classes of lactose mutants. The ONPG technique allowed them to determine if the genes enabling lactose to be used were transcribed and translated even if lactose was not present in the medium. Observe colonies grown on a medium containing a carbon and energy source, such as glycerol, but not containing lactose. When ONPG is applied, a few of these colonies will turn yellow. The regions of the lac operon, which could be mutated to yield this result, are lac-I promoter, lac-I, and operator. If the lac-I gene is mutated so that the repressor is not expressed or is non-functional, the repressor will not be able to bind to the operator site. Therefore, in the presence or absence of lactose, the structural genes will be expressed, and the yellow indicator color will appear. If the operator site is mutated, the repressor will not be able to bind. Therefore, in the presence or absence of lactose, the structural genes will be expressed, and the yellow indicator color will appear. Let's observe the role of glucose in the expression of the lac structural genes. E. coli cells always produce
I hate how it's
enzymes
talking.
for
E.
glucose
coli
breakdown.
cells.
So, if glucose is abundant in the environment, E. coli will use this sugar as an energy source and repress the expression of the lac operon. This adaptation saves energy by reducing the production of unnecessary enzymes, those of the lac operon. This form of inhibition is known as catabolite repression. What happens to the lac operon in the absence of glucose? An enzyme called adenylyl cyclase is continually active in converting ATP molecules into cyclic AMP.
Okay, wait, hold on. That was the part.
An enzyme called adenylyl cyclase is continually active in converting ATP molecules into cyclic AMP. Cyclic AMP molecules. A pyrophosphate ion is a byproduct of each reaction. Under these conditions, the cell produces a pool of cyclic AMP molecules. Cyclic AMP molecules bind to protein.
Okay, so we found the answer, though. It's adenylyl cyclase.
Called catabolite activator proteins, or CAPs for short. When a CAP binds to a cyclic AMP, it can then bind to a site near the lac operon's promoter, called the CAP site. CAP facilitates a functional interaction of RNA polymerase with the promoter and thereby stimulates transcription. This is an example of positive control, in which the binding of a protein, in this case CAP, activates transcription. Note that lactose must also be present in the cell. Otherwise, the lac repressor would bind to the operator site and block transcription. When glucose is present in the environment and transported into the cell, the function of adenylyl cyclase is inhibited. The result is that ATP is no longer converted into cyclic AMP.
Okay, so glucose inhibits the production of cyclic AMP, and then cyclic AMP positively impacts. And then what did they?
Cyclic AMP pool drops. Without cyclic AMP, CAP cannot bind to the CAP site and attract RNA polymerase to the lac promoter.
Polymerase.
Thus, in the presence of glucose, the lac operon is not transcribed efficiently. Glucose and lactose both affect the transcription of the lac structural genes. E. coli lives more efficiently because it can regulate expression of the lac operon based on the presence or absence of glucose and lactose. Which environmental condition would produce the arrangement of proteins on the lac operon shown here and result in no transcription of the lac structural genes?
Wait, wait, we gotta find that part that answers this one.
Finding PG tech were source ONP, the regions of the guidinal gene structural lacs for glucose. Let's observe the role of glucose in the expression of the lac structural genes. E. coli cells always produce the enzymes for glucose breakdown. So, if glucose is abundant in the environment, E. coli will use this sugar as an energy source and repress the expression of the lac operon. This adaptation saves energy by reducing the production of unnecessary enzymes, those of the lac operon. This form of inhibition is known as catabolite repression.
Okay.
The lac operand in E. coli.
Bro, you gotta be kidding me.
Bacteria adapt to changes in their surroundings by using regulatory proteins to
How
turn groups
much
of
are we
genes
gonna
on and
do
off in
this
response
thing?
to various environmental signals. The DNA of Escherichia coli is sufficient to encode about 4,000 proteins, but only a fraction of these are made at any one time. E. coli regulates the expression of many of its genes according to the food sources that are available to it. In 1961, Francois Jacob and Jacques Monod proposed the operand model of gene regulation in bacteria. The model was based on their study of the genes in E. coli that code for enzymes that affect the breakdown of lactose. Because of the pioneering work of Jacob and Monod, the lac operand is typically used to illustrate gene regulation in bacteria. An operand is cluster of bacterial genes along with the adjacent promoter that controls the transcription of those genes. When the genes in an operand are transcribed, a single mRNA is produced for all the genes in an operand. The promoter contains short, specific nucleotide sequences that signal the start of a gene, and the site where RNA polymerase starts transcription. Operators are regions of DNA that interact with regulatory proteins that control the transcription of operands. The regulatory gene of the lac operand produces an mRNA that produces a repressor protein, which can bind the operator of the lac operand. The general term for the product of a regulatory gene is a regulatory protein. The lac regulatory protein is called a repressor because it keeps RNA polymerase from transcribing structural genes. Thus, the repressor inhibits transcription of the lac operand. When lactose is present, it binds to the repressor and changes its shape. As a result of this change, the repressor can no longer bind to the operator region. RNA polymerase can then bind to the promoter and
I
transcribe
mean,
lactose.
why am I even watching this? It's like, come on. I mean, like, come on, dude. These homeworks are so stupid. I mean, like, oh, jeez. Which of these is a regulatory E? Wait, is the regulatory gene? Oh. I don't understand what this is asking. A longer term response. How are the processes shown in the figure below examples of feedback mechanisms regulating biological systems in bacterial cells? Turns off expression of genes encoding pathway enzymes. Necessary for the synthesis regulation of compounds. I don't even know what this is fucking asking. A longer term response? What does that even mean? There's no hint on this, dude. These homeworks are so dumb. Like, what the hell does that even mean? No hint, like, what is this even fucking asking, dude? I'm pretty sure that inhibition by the final product has to be both because the arrow goes to both. Does anyone have the answer for this? I turned up my phone. Fuck. This one is so... Do I have to go through this whole thing again? Oh, thank God, no. Like, what is this asking, dude? Okay, so what they're saying, regulation of enzyme activity is necessary for the synthesis regulation of compounds that bacteria are able to. I already did this one. This is incorrect. It's like, this question is so stupid. I think we should try to switch this and this. Regulation of enzyme production turns off expression of genes. That's true. I don't know about the longer term response thing, to be honest. Maybe this is neither. I feel like this is wrong. Bro. Inhibition by the final product is definitely both. How many tries do we get? This is what I thought, but I think this is still wrong, right? Oh my god, this problem is so stupid. This is a move on. Fuck, that's so dumb. There's a group of genes in the genome of the described bacterium called Lux genes, which are organized in an operon, reaching a certain concentration. Bro, what? Molecules involved in this process activate the Lux genes. I feel like that's got to be what it is. As a result, the population of bacteria needs to reach an There's not even... Is there a hint? Of course not. Right? Okay, that's what I think. Activate, activate, activate. Okay, good. God, I hate these homeworks. They're so stupid. Why is the lac operon said to be inducible? If a particular operon encodes enzymes for making an essential amino acid and is regulated with a trip operon, then... What would occur if the repressor of an inducible operon were mutated so it could not bind the operator? It would be on. Continuous transcription of the operon. Which statement about inducible operons are correct? In an inducible operon, an inducer inactivates the repressor. In an inducible operon, the repressor is synthesized in its active form. Inducible enzymes generally function in synthetic pathways that produce end products from raw material. Repressible operants usually contain genes that code for anabolic. Okay, I'm just gonna Okay, so someone already asked and nobody responded after like 3 days. So we're just gonna freaking spam answers. So this is true. I don't know about this one. This one, they're both, I mean, those got to be true. Turns off expression of genes, that's got to be this one. So now I would assume this is longer term response. I mean, like, how many times do we get to just do this thing? Oh, okay, I'm out of times. Wait, what? Okay, you know what? I don't fucking care. It's already gone. It's done. But this directly contradicts this. I don't care. All right, we're done with that one. I'm gonna go make a sandwich or something. Apparently, there's like a free food somewhere, but I don't know. Part of the RNA polymerase itself specifically recognizes and binds the promoter. Bacteria, part of the RNA polymerase itself specifically. Well, that's actually kind of crazy. They gotta like catch up to it. That is freaky. I feel like eukaryotes like it's like a messy code base. It's like what the hell? That's how you got that thing to work? If it works, it works, I guess. No changing it now. For real. No way you're gonna change how that happens. What the 3' end receives a 5' cap. This is not the 3' end though. I think they meant to write 5'. I'm pretty sure that's just an error in the book. Dude, what? The introns are longer? Whoa. What the? That's crazy. 20 times. Yeah, that's fine. Oh. Oh, okay. All right, that actually makes sense. All right. Yeah. It's not that, because it's not one codon to one amino acid. It's like one codon to like a thousand amino acids, or I don't know how many, but yeah, that definitely makes sense. Dude, this intron and exon thing is wild. The 5' splice site at the start of the intron. These things seem so fuzzy. This response is a little too technical for me, but this is interesting. I'm glad I asked that because it's like, yeah, maybe they're equivalent extension costs, but they're not. There's an amplification that you don't think about. This complex binds to several short nucleotide sequences along an intron. Whoa. Oh, yeah. I mean, when you think purely informational about the major, specifically the nucleic acids, it's like, okay, but, those bases are. One important consequence of the presence of introns in genes is that a single gene can encode more than one kind of polypeptide. Many genes are known to give rise to two or more different polypeptides, depending on which segments are treated as exons during RNA processing. This is called alternative RNA splicing. Uh, the HGP suggests that alternative RNA splicing is one reason humans can get along with about the same number of genes as a nematode. Because of alternative splicing, the number of different protein products an organism produces can be much greater than the number of its genes. Proteins often have a modular architecture consisting of discrete structural and functional regions called domains. Okay. Wait, what's happening here? Why is it not? Is there two mics in there? That doesn't seem right. I think it's mono. We have to change the infill. We'll just see. I don't even know what I have in there. All right, so this is the part that I got like all the questions wrong on in my exam, I think. Translation. All right, I gotta read it out loud because I'm losing focus. In the process of translation, a cell reads a genetic message and builds a polypeptide accordingly. The message is a series of codons along an mRNA molecule, and the translator is called tRNA. The function of a tRNA is to transfer an amino acid from the cytoplasmic pool of amino acids to a growing polypeptide in a ribosome. A cell keeps its cytoplasm stocked with all 20 amino acids, either by synthesizing them from other compounds or by taking them up from surrounding solution. The ribosome, a structure made of proteins and RNAs, adds each amino acid brought to it by a tRNA to the growing end of a polypeptide chain. Translation is simple in principle, but complex in its biochemistry and mechanics, especially in the eukaryotic cell. In dissecting translation, we'll focus on the slightly less complicated version of the process that occurs in bacteria. We'll first look at the major players in this process, the structure and function of transfer RNA. The key to translating a genetic message into a specific amino acid sequence is the fact that each tRNA molecule enables translation of a given mRNA codon into a certain amino acid. This is possible because tRNA bears a specific amino acid at one end of its three-dimensional structure, while at the other end is a nucleotide triplet that can base pair with the complementary codon on mRNA. A tRNA molecule consists of a single RNA strand that is only about 80 nucleotides long, compared to hundreds of nucleotides for most mRNA molecules. The presence of complementary stretches of nucleotide bases that can hydrogen bond to each other allows this single strand to fold back on itself and form a molecule with a particular 3D structure. Flattened into one plane to clarify this base pairing, a tRNA molecule looks like a clover leaf. Is that what that looks like? The tRNA actually twists and folds into a compact 3D structure that is roughly L-shaped, with the 5' and 3' ends of the linear tRNA located with the 5' and 3' ends of the linear tRNA both located near one end of the structure. The protruding 3' end acts as an attachment site for an amino acid. The loop extending from the other end of the L includes the anticodon, the particular nucleotide triplet that base pairs to a specific mRNA codon. Thus, the structure of a tRNA molecule fits its function. The anticodons are conventionally written 3' to 5' to align with properly with the codons written 5' to 3'. For base pairing, RNA strands must be anti-parallel, like DNA. As an example of how tRNAs work, consider the mRNA codon GGC, 5' GGC 3', which is translated as the amino acid glycine. And I think glycine is the simplest one. Yeah, glycine is the simplest, yeah, because it's just a H as its R group. I wonder what it. Yeah, can we go to AlphaFold? Can I just submit something right now? Search for a gene. What if I just did that? Let's just do the smallest. Can it fold just a chain of glycine? Okay. The tRNA that base pairs with this codon by hydrogen bonding has 3-CCG5 as its anticodon and carries glycine at its other end. So the glycine goes here, I guess. Yeah. As an mRNA molecule is moved through a ribosome, glycine will be added to the polypeptide chain whenever the codon 5GGC3 is presented for translation. Codon by codon, the genetic message is translated as tRNAs position each amino acid in the order prescribed, and the ribosome adds that amino acid on to the growing polypeptide chain. The tRNA molecule is a translator in the sense that in the context of the ribosome, it can read a nucleic acid word and interpret it as a protein word. Like mRNA and other types of cellular RNA, transfer RNA molecules are transcribed from DNA templates. In a eukaryotic cell, tRNA, like mRNA, is made in the nucleus and then travels to the cytoplasm, where it will participate in the process of translation. In both bacterial and eukaryotic cells, each tRNA molecule is used repeatedly, picking up its designated amino acid in the cytosol, depositing this cargo onto the polypeptide chain at the ribosome, and then leaving the ribosome, ready to pick up another of the same amino acid. The accurate translation... So, I mean, it seems like one of those things that probably has a relatively low production rate and very stable. Well, I don't know. Maybe like the global protein synthesis demand varies a lot, or maybe it varies a little. I don't know. Oh, really? Okay, now let's do something bigger. So let's do something 10 times bigger. So now we're going to go to... What? I can't paste in here? That is ridiculous. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Is it just going to get longer and longer? And the next one we'll do will be 100, and then we'll do 200, and then we'll do 400. I have enough jobs for it, so... The cofactor thing also seems pretty interesting. I remember looking into that. The accurate translation of a genetic message requires 2 instances of molecular recognition. First, a tRNA that binds to an mRNA codon specifying a particular amino acid must carry that amino acid, and no other, to the ribosome. The correct matching up of tRNA and amino acid is carried out by a family of related enzymes that are aptly named amino acyl tRNA synthetases. The active site of each type of amino acyl tRNA synthetase fits only a specific combination of amino acid and tRNA. Regions of both the amino acid attachment end and the anticodon of the tRNA ensure the specific fit. So, like, let's look at this. Okay, so yeah, so it's got to be big because it only is going to fit these 2 active sites basically. Is it done already? Oh, really? Nah, no way, dude. That's sick. If folded up, it forms loops. Okay, all right, let's do 100 now. Okay, so do I still have this? So this is 80. Let's just do 120. We can fit so many fucking glycines in this bitch. You know, like, what is it that makes it bend around? Like, AlphaFold must have some sort of world model of, like, the, you know, it must have learned some information about the cell environment, because otherwise, why would it not just polymerize linearly indefinitely? There must be some constraint on protein length that it's like encoded or something like that. So now here we're getting kind of this bent. You know, it's a really oily, you know, hydrophobic. I mean, the whole thing is extremely low confidence, isn't it? For some reason, it seems to me that, like, it would be a good experiment to. It seems like it would be a good experiment to basically see how it how it crystallizes, right? Is that the right word? Homopolymer. That's fun. All right. Enough of that. Let's get back to the book. Let's just look at this diagram. So the RNA so there's a synthetase for all 20. Wait. There are 20 different synthetases, one for each amino acid. A synthetase joins a given amino acid to the appropriate tRNA. One synthetase is able to bind to all of the different tRNAs for its particular amino acid. I don't get that comment. The synthetase catalyzes the covalent attachment of the amino acid to its tRNA in a process driven by the hydrolysis of ATP. The resulting amino acid tRNA, also called a charged tRNA, is released from the enzyme and is then able to deliver its amino acid to the growing polypeptide chain on a ribosome. The second instance of molecular recognition is the pairing of the tRNA anticodon with the appropriate mRNA codon. If one tRNA type existed for each mRNA codon, there would be 61. Why not 64? In bacteria, however, there are only about 45 tRNAs, signifying that some tRNAs must be able to bind to more than one codon. This is possible because the rules for base pairing between the third nucleotide base of a codon and the corresponding base of a tRNA anticodon are relaxed compared to those at other codon positions. For example, the nucleotide base U at the 5' end of a tRNA anticodon can pair with either A or G in the third position. This flexible base pairing at this codon position is called wobble. Wobble explains why the synonymous codons for a given amino acid most often differ in the third nucleotide base. Accordingly, a tRNA with the anticodon UCU can base pair with either the mRNA AGA or AGG. Both of which code for arginine. The structure and function of ribosome. How much more do we have on this chapter? A whole lot. Yeah, we're going to skip the mutations thing, but we'll read to 356, and then I'll go on to 18. The structure and function of ribosomes. Although the ribosomes of bacteria and eukaryotes are very similar in structure and function, eukaryotic ribosomes are slightly larger and differ somewhat from bacterial ribosomes in their molecular composition. The differences are medically significant. Certain antibiotic drugs can inactivate bacterial ribosomes without affecting the ability of eukaryotic ribosomes to make proteins. These drugs, including tetracycline and streptomycin, are used to combat bacterial infections. Ribosomes facilitate the specific coupling of tRNA anticodons with mRNA codons during protein synthesis. A ribosome consists of a large subunit and a small subunit, each made up of proteins and one or more ribosomal RNAs. In eukaryotes, the subunits are made in the nucleolus. Ribosomal RNA genes are transcribed, and the RNA is processed and assembled with proteins imported from the cytoplasm. Completed ribosomal subunits are then exported via nuclear pores to the cytoplasm. In both... Wait. Yeah, okay. In both bacteria and eukaryotes, a large and small subunit join to form a functional ribosome only when attached to an mRNA molecule. About 1/3 of the mass of a ribosome is made up of proteins. The rest consists of three rRNA molecules, or 4 in eukaryotes. Because most cells contain thousands of ribosomes, RNA is the most abundant type of cellular RNA. By a lot, too. It's not even it's not a little bit, it's a lot. The structure of a ribosome reflects its function of bringing an mRNA molecule together with tRNA carrying amino acids. The mRNA itself has a binding site for the ribosome. The ribosome, in turn, has a binding site for mRNA, as well as 3 binding sites for tRNA. The P site holds the tRNA carrying the growing polypeptide chain. The A site holds the tRNA containing the next amino acid to be added to the chain. Discharged tRNAs leave the ribosome for the E site, the exit site. The ribosome holds the tRNA and mRNA in close proximity and positions the new amino acid so that it can be added to the carboxyl end of the growing polypeptide. All right, so it goes N to C when it gets added. It then catalyzes the formation of the peptide bond. As the polypeptide becomes longer, it passes through an exit tunnel in the ribosome's large subunit. When the polypeptide is complete, it is released through the exit tunnel. The widely accepted model is that rRNAs, rather than ribosomal proteins, are primarily responsible for both the structure and function of the ribosome. The proteins, which are largely on the exterior, support the shape changes of the rRNA molecules as they carry out catalysis during translation. Ribosomal RNA is the main constituent of the A and P sites and of the interface between the two subunits. It also acts as a catalyst of peptide bond formation. It also acts as the catalyst for of peptide bond formation. Thus, a ribosome could actually be considered one colossal ribozyme. Building a polypeptide. We can divide translation, the synthesis of a polypeptide, into 3 stages: initiation, elongation, and termination. All 3 require protein factors that aid in the translation process. Some steps of initiation and elongation also require energy, provided by the hydrolysis of guanosine triphosphate. Is it after guano? Yeah. We got G is named after poo. That's fun. All right. Ribosome association and initiation of translation. The initiation stage of translation brings together an mRNA, tRNA bearing the first amino acid of the polypeptide, and the two subunits of a ribosome. First, a small ribosomal subunit binds to the mRNA and a specific initiator, tRNA. I got that one wrong on the test. Which carries the amino acid methionine. In bacteria, the small subunit can bind the two in either order. In bacteria, the small subunit can bind the two in either order. It binds the mRNA at a specific RNA sequence just upstream of the start codon. In eukaryotes, the small subunit with the initiator tRNA already bound binds to the 5' cap of the RNA, then moves or scans downstream along the mRNA until it reaches the AUG start codon, where the initiator tRNA then hydrogen bonds, as shown as step 1 of. In a bacterial cell, the mRNA binding site on the subunit recognizes a specific what? Okay, so then the arrival of a large ribosomal subunit completes the initiation complex. Proteins called initiation factors not shown are required to bring all the translation components together. Hydrolysis of GTP provides the energy for the assembly. The initiator tRNA is in the P site. The A site is available to the tRNA bearing the next amino acid. The union of mRNA, initiator tRNA, and the small ribosomal subunit is followed by the attachment of a large ribosomal subunit, completing the translation initiation complex. Proteins called initiation factors are required to bring all these components together. The cell also expends energy obtained by hydrolysis of a GTP molecule to form the initiation complex. At the completion of the initiation process, the initiator tRNA sits in the P site of the ribosome, and the vacant A site is ready for the next amino acyl tRNA. Note that a polypeptide is always synthesized in one direction, from the initial methionine at the amino end, called the N-terminus, towards the final amino acid at the carboxyl end, called the C-terminus. Elongation of the polypeptide chain. In the elongation stage of translation, amino acids are added one by one to the previous amino acid at the C-terminus of the growing chain. Each addition involves several proteins called elongation factors and occurs in a 3-step cycle. Energy expenditure occurs in the first and third steps. Codon recognition requires hydrolysis of... Oh, really? Codon recognition requires hydrolysis of one molecule of GTP, which increases the accuracy and efficiency of this step. One more GTP is hydrolyzed to provide energy for the translocation step. The mRNA is moved through the ribosome in one direction. 5' end first. This is equivalent to the ribosome moving 5 to 3 on the mRNA. Yeah, okay. The main point is that the ribosome and the mRNA move relative to each other, unidirectionally codon by codon. The elongation cycle takes less than 1/10 of a second in bacteria and is repeated as each amino acid is added until the polypeptide is completed. The empty tRNAs that are released from the E site return to the cytoplasm where they'll be reloaded with the appropriate amino acid. Termination of translation. The final stage of translation is termination. Elongation continues until a stop codon in the mRNA reaches the A site. I think I might have got that question right on the exam. Elongation continues until a stop codon in the mRNA reaches the A site. The nucleotide-based triplets UAG, UAA, and UGA, all written 5-3, do not code for amino acids but instead act as signals to stop translation. A release factor, a protein shaped like an amino acid tRNA, binds directly to the stop codon. Is there a picture? This is the elongation process. Here's the termination process. All right, so we got a special... When a ribosome reaches a stop codon on an mRNA, the A site of the ribosome accepts a release factor, a protein shaped like a tRNA instead of an amino acid tRNA. The release factor promotes hydrolysis of the bond between the tRNA and the P site and the last amino acid in the polypeptide, thus freeing the polypeptide from the ribosome. The two ribosomal subunits and the other components of the assembly dissociate with the hydrolysis of two GTP molecules. I want to see this translocation. How does this work? The ribosome translocates the tRNA in the A site to the P site. At the same time, the empty tRNA in the P site is moved to the E site where it is released. The mRNA moves along with its bound tRNAs, bringing the next codon to be translated into the A site. Okay. Completing and targeting the functional protein. The process of translation is often not sufficient to make a functional protein. In this section, you will learn about modifications that polypeptide chains undergo after translation, as well as some of the mechanisms used to target completed proteins to specific sites in the cell. Okay. Okay. Protein folding and post-translational modifications. During its synthesis, a polypeptide chain begins to coil and fold spontaneously as a consequence of its amino acid sequence primary structure. Forming a protein with a specific shape, a three-dimensional molecule with secondary and tertiary structure, forming a protein with a specific shape. Thus, a gene determines primary structure, in which determines shape. Okay. All right, I think we get all this. All right. Additional steps. Post-translational modifications may be required before the protein can begin doing its particular job in the cell. Certain amino acids may be chemically modified by the attachment of sugars, lipids, phosphate groups, or other additions. Enzymes may remove one or more amino acids from the leading amino and N-terminus of the polypeptide chain. They call that the leading? In some cases, a polypeptide chain may be enzymatically cleaved into two or more pieces. In other cases, two or more polypeptides that are synthesized separately may come together. If the protein is quaternary structure, an example is hemoglobin. Yeah. Quaternary. Targeting polypeptides to specific locations. In electron micrographs of eukaryotic cells active in protein synthesis, two populations of ribosomes are evident: free and bound. Free ribosomes are suspended in the cytosol and mostly synthesize proteins that stay in the cytosol and function there. In contrast, bound ribosomes are attached to the cytosolic side of the ER or to the nuclear envelope. Bound ribosomes make proteins of the endomembrane system, as well as proteins secreted from the cell, such as insulin. It is important to note that the ribosomes themselves are identical and can alternate being between free and bound. What determines whether a ribosome is free in the cytosol or bound to rough ER? Polypeptide synthesis always begins in the cytosol as a free ribosome starts to translate an mRNA molecule. There, the process continues to completion unless the growing polypeptide itself cues the ribosome to attach to the ER. The polypeptides of proteins destined for the endomembrane system or for secretion are marked by a signal peptide, which targets the protein to the ER. Okay, polypeptide synthesis become free. Wow. An SRP, signal recognition particle, binds to the signal peptide, if present, halting synthesis momentarily. Oh, interesting. Okay, translocation complex. The SRP binds to the receptor protein in the ER membrane, part of a protein complex that forms a pore. Oh, really? This is super interesting. The SRP leaves, and the polypeptide synthesis resumes with simultaneous translocation across the membrane. The signal peptide is cleaved by an enzyme. Is this only in, yeah, this has to be only in eukaryotes. The rest of the polypeptide leaves the ribosome and folds into its final conformation. Okay. The signal peptide, a sequence of about 20 amino acids at or near the leading N-terminus of the polypeptide, is recognized as it emerges from the ribosome by a protein RNA complex called an SRP, signal recognition particle. This particle escorts the ribosome to a receptor protein built into the ER membrane. The receptor is part of a multi-protein translocation complex. Polypeptide synthesis continues here, and the growing polypeptide snakes across the membrane into the ER lumen via protein pore. The signal peptide is removed by an enzyme. The rest of the completed polypeptide, if it is to be secreted from the cell, is released into solution within the ER lumen. Or if the polypeptide is to be a membrane protein, amino acid sequences further in the chain cause that part to remain embedded in the ER membrane. In either case, it travels in a transport vesicle to its destination. Other kinds of signal peptides are used to target polypeptides to mitochondria, chloroplasts, the interior of the nucleus, and other organelles that are not part of the endomembrane system. The critical difference in these cases is that translation is completed in the cytosol before the polypeptide is imported into the organelle. Translocation mechanisms also vary, but in all cases studied to date, the postal zip codes that address proteins for secretion or to cellular locations are signal peptides of some sort. Bacteria also employ signal peptides to target proteins to the plasma membrane or for secretion. Interesting. What chapter is this? Making multiple polypeptides in bacteria and eukaryotes. In previous sections, you learned how a single polypeptide is synthesized using the information encoded in an mRNA molecule. When a polypeptide is required in a cell, though, the need is for many copies, not just one. In both bacteria and eukaryotes, multiple ribosomes translate an mRNA at the same time. That is, a single mRNA is used to make many copies of a polypeptide simultaneously. Once a ribosome is far enough past the start codon, a second ribosome can attach the mRNA, eventually resulting in a number of ribosomes trailing the mRNA along the mRNA. Such strings of ribosomes, called polyribosomes or polysomes, can be seen with an electron microscope. They can be either free or bound. They enable a cell to rapidly make many copies of a polypeptide. Another way both bacteria and eukaryotes increase the number of copies of a polypeptide is by transcribing multiple mRNAs from the same gene. However, the coordination of the two processes, transcription and translation, differs in the two groups. The most important differences between bacteria... Oh wow, that's cool. That's really neat. That is really freaking cool. The most important differences between bacteria and eukaryotes arise from the bacterial's lack of cell's lack of compartmental organization. Like a one-room workshop, a bacterial cell ensures a streamlined operation by coupling the two processes. With no nuclear envelope, it can simultaneously transcribe and translate the same gene, and the newly made protein can quickly diffuse to its site of function. In contrast, the eukaryotic cell nuclear envelope segregates transcription from translation and provides a compartment for extensive RNA processing. This processing stage includes additional steps discussed earlier, the regulation of which can help coordinate the eukaryotic cell's elaborate activities. Okay, we're skipping mutations, and then we're going to skip the gene editing thing. I mean, it seems interesting, but we just don't have time. It's only a page. Okay, I'll read it out loud. Ever since biologists have understood how disease-causing proteins were the result of mutations in genes, they have sought techniques for gene editing, altering genes in a specific, predictable way. Their aim has been to use such a technique to change specific genes in living cells, in part to try to correct genes that cause disease. Over the past 15 years, biologists have developed a powerful new technique for gene editing called the CRISPR-Cas9 system that is transforming the field of genetic engineering. The direct manipulation of genes for practical purposes. Cas9 is a bacterial protein that helps defend bacteria against bacteriophages. In bacterial cells, Cas9 acts together with a guide RNA made from the CRISPR region of the bacterial genome. Similar to other DNA cutting enzymes involved in DNA repair, Cas9 is a nuclease that cuts double-stranded DNA molecules. The power of Cas9 for gene editing is that the Cas9 protein will cut any sequence to which it is directed. Cas9 is directed to its target by a guide RNA molecule that it binds and uses as a homing device, cutting both strands of any DNA sequence that is complementary to the guide RNA. Scientists have been able to exploit the function of Cas9 by introducing the Cas9 guide RNA complex into a cell they wish to alter. The guide RNA in the complex is engineered to be complementary to the target gene. Cas9 cuts both strands of the target DNA, and the resulting broken ends of a DNA trigger a DNA repair system. When there is no undamaged DNA for the enzymes of the repair system to use as a template, the repair enzymes introduce or remove random nucleotides while rejoining the ends. Generally, this process alters the DNA sequence so that the gene no longer works properly. This technique is a highly successful way for researchers to knock out and disable a given gene to study what the gene does in an organism. But what about using this system to help treat genetic diseases? Researchers have modified the technique so that CRISPR-Cas9 can be used to repair a gene that has a harmful mutation. They introduce a segment of the normal functional gene along with the CRISPR-Cas9 system. After Cas9 cuts the target DNA, repair enzymes use the normal DNA as a template to repair the target DNA at the breakpoint. In this way, the CRISPR-Cas9 system edits the defective gene so that it is corrected. In 2018, researchers reported promising results using to attempt to correct the sickle cell. Oh, really? I didn't know that they tried it on sickle cell. But and then Huntington's recently was the big one. Okay. So now we're doing regulation of. I think we probably want to just jump to the. Yeah, we know all the bacterial gene regulation stuff is pretty straightforward, I think. So I'm gonna skip that. But I mean, I'm just trying to finish this freaking homework. But I don't know anything about this chromatin stuff. Hopefully this one will be relatively short, but I don't know. Eukaryotic gene expression is regulated at many stages. I'm actually excited to read about this because this is pretty interesting stuff. All organisms, whether prokaryotes or eukaryotes, must regulate which genes are expressed at any given time. Both unicellular organisms and the cells of multicellular organisms continually turn genes on and off in response to signals from their external and internal environments. Regulation of gene expression is also essential for cell specialization in multicellular organizations, which are made up of different types of cells. To perform its own distinctive role, each cell type must maintain a specific program of gene expression in which certain genes are expressed and others are not. Differential gene expression. A typical human cell might express about a third to a half of its protein coding genes at any given time. Highly differentiated cells, such as muscle or nerve cells, express a smaller fraction of their genes. Almost all of the cells in a multicellular organism contain an identical genome. Cells of the immune system are one exception. Oh, that's really crazy. What's chapter 43 about? Yeah, we never did this in 6A. We never did immune system. Damn, that's actually sad. I want to read that one. I don't know anything about the immune system. That's super cool. A subset of genes is expressed in each cell type. Some of these, about 35%, are housekeeping genes expressed by many cell types, while others are unique to that cell type. The uniquely expressed genes allow these cells to carry out their specific function. The differences between cell types, therefore, are due not to different genes being present, but to differential gene expression, the expression of different genes by cells with the same genome. The function of any cell depends on its expressing the appropriate set of genes. The transcription factors of a cell must locate the right genes at the right time, a task like finding a needle in a haystack. Abnormal gene expression can cause serious imbalances and diseases, including cancer. Figure 18.6 summarizes the process of gene expression in a eukaryotic cell, highlighting the key stages in the expression of a protein-coding gene. Many genes have more than one control point. Okay. When the structure of DNA was determined in 1953, an understanding of the mechanisms that control gene expression in eukaryotes seemed almost hopelessly out of reach. Since then, advances in DNA technology have enabled molecular biologists to uncover many details of eukaryotic gene expression. In all organisms, gene expression is commonly controlled at transcription. Regulation at this stage often occurs in response to signals coming from outside the cell, such as hormones. I forget hormones. Okay, yeah. So hormones typically control gene regulation. For this reason, the term gene expression is often equated with transcription for both bacteria and eukaryotes. While this may be the case for bacteria, the greater complexity of eukaryotic structure, cell structure, and function provides opportunities for regulating gene expression at many stages besides just transcription. Let's examine some of the important control points of eukaryotic gene expression more closely. All right, here we go. We're finally getting into the stuff that I need to do for my homework. Regulation of chromatin structure. Recall that the DNA of eukaryotic cells is packaged with proteins in an elaborate complex known as chromatin, the basic unit of which is the nucleosome. Mm. Okay. The structural organization of chromatin not only packs a cell's DNA into a compact form that fits inside the nucleus, but also helps regulate gene expression in several ways. Genes within heterochromatin, which is more densely arranged than euchromatin, are usually not expressed. In euchromatin, whether or not a gene is transcribed is affected by the location of a nucleosomes along a gene's promoter. The location of nucleosomes along a gene's promoter, and also to the sites where the DNA attaches to the protein scaffolding of the chromosome. And also the sites where DNA attaches to the proteins. Okay. Chromatin structure and gene expression can be influenced by chemical modifications of both the histone proteins of the nucleosomes around which DNA is wrapped and the nucleotides that make up the DNA. Here we examine the effects of these modifications, which are catalyzed by enzymes Okay. Histone modifications and DNA methylation. Chemical modifications to histones found in all eukaryotic organisms play a direct role in the regulation of gene transcription. The N-terminus of each histone protein in a nucleosome protrudes outwards from the nucleosome. These so-called histone tails are accessible to various modifying enzymes that catalyze the addition or removal of specific chemical groups such as acetyl, methyl, and phosphate. Generally, histone acetylation, the addition of an acetyl group to an amino acid in the histone tail, appears to promote transcription by opening up chromatin structure. But how does it know where to go? While the addition of methyl groups to histones can lead to the condensation of chromatin and reduce transcription, often the addition of a particular chemical group may create a new binding site for enzymes that can further modify chromatin structure. Rather than modifying histone proteins, a different set of enzymes can methylate the DNA itself on certain bases, usually cytosine. Such DNA methylation occurs in most plants, animals, and fungi. Long stretches of inactive DNA, such as that of inactivated mammalian X chromosomes, are generally more methylated than regions of actively transcribed DNA. On a smaller scale, the DNA of individual genes is usually more heavily methylated in cells in which the DNA of individual genes is usually more heavily methylated in cells in which those genes are not expressed. Removal of the extra methyl groups can turn on some of these genes. Epigenetic inheritance. The chromatin modifications that we just discussed do not change the DNA sequence, yet they still may be passed along to future generations of cells. Inheritance of traits transmitted by mechanisms not involving the nucleotide sequence itself is called epigenetic inheritance, the study of which is called epigenetics. Whereas mutations in the DNA are permanent changes, modifications to the chromatin can be reversed. For example, DNA methylation patterns are largely erased during gamete formation and reestablished during embryonic development. Furthermore, they are changeable, thus responding more rapidly to environmental conditions. Research on epigenetics has skyrocketed over the past 20 years. The importance of epigenetic information in regulating gene expression is now widely accepted. One key study used a mouse mutant whose genome had been altered so that a gene called a Gaudi, that determines coat color had normally expressed only briefly during fur formation, was instead expressed throughout development. This overexpression resulted in yellow mice rather than the previous work had shown that merely supplementing the diet of pregnant mothers with methyl group containing compounds like folic acid. I think folic acid. Is it folic acid? Yeah. LA Taco. It's colonizing all over again. Oh, that's funny, man. Rattling LA's taco universe. I gotta read this. I mean, obviously, this is a stupid law. I mean, okay, I'm actually not going to say that. I retract that. It's not a stupid law. It's just one of those cases where the government is using force as a tool for awareness. Like, nobody knows about folic acid. Nobody knows about the defects. You know, I don't think very many people know about this. Yeah, that's right. Neural tube. Neuralation, bro. The bill's finding and declaration section includes statements asserting a disproportionately higher risk and higher rate of NTDs in Latino communities, but these are presented without supporting evidence numbers. Really? Okay, that's dumb. Let's look up folic acid. What's its pKa? What? It's an acid with no pKa? Advocates celebrate the bill as a win for fortified food equality. Since the initial drug, because I think they put it in bread like in the 90s. Oh yeah, here you go, grain. Yeah, that's what I thought. Oh yeah, bro, I can't even read. Yeah, I mean, this was like when I first read this, like before the tortilla stuff. Like I was like, that's actually pretty cool. Like that's actually pretty cool. What is nixtamal? Yeah, it's kind of got a similar pK to acetic acid. Hmm. Not that I know what that even means. Bro's literally 3 weeks into OChem and thinks he knows all the pKa's. Yeah, I do. I think that I really don't think that it's necessary, but like, uh, you know, I wouldn't like. How do you get people to know? And also, why folic acid? Like, why does that prevent neural tube defects? Yeah, here you go. I mean, this is the diagram that you see in. Well, actually, this is a little bit different. Oh, but this is. Oh, God, that's horrible. What is the mechanism of action? Wow, this has been studied a lot. Okay, defects per million. It's pretty low, but it's also like somehow we figured out like all right, well we got to keep going. I mean, I could go all day into this, but I just thought it was a fun folic acid shout out. All right, so previous work had shown that merely supplementing the diet of pregnant mothers with methyl group containing compounds like folic acid could shift the range of coat colors of the offspring back to normal. Feeding methyl groups to mothers, why don't we just feed them gasoline? Or, methane. Or ethane probably is even better if you could cleave ethane. Man, I forgot all this development stuff.
Day 16. The embryo consists of 3 layers: endoderm, mesoderm, and ectoderm. Ectoderm develops into the nervous system, and the first phase of this process is known as neurulation. Around day 18, part of the ectoderm begins to thicken. This portion is known as the neural plate, in dark blue, between the cranial end of the embryo and the primitive node. Neurulation is the process of neural plate becoming neural tube. Got a schematic animation here to try and illustrate that process. We'll break this down day by day throughout the rest of the video. On day 19, the neural plate begins to buckle in the midline. We call this dip the neural groove, and the mounds either side the neural folds. These folds progress toward each other and begin to fuse around day 21. Where they have fused, they form a hollow cylinder, which is the neural tube. Fusion then continues in synchrony toward caudal and cranial ends.
Caudal. I haven't heard
The
that
as yet
word in a long
unfused
time,
portions are known respectively as the cranial neuropore and caudal neuropore. Pore literally means minute opening. The minute opening at the cranial end closes around day 24, and the caudal end closes around day 26. A particular group of cells reside at the peaks of the neural folds. These are the neural crest cells, which form a layer over the top of the closed neural tube. These develop into most of the peripheral nervous system. To sum up, neurulation is the process of neural plate becoming neural tube. The closure of the neural folds begins in the center and zips up towards either end. The central nervous system is developed from the neural tube. It's worth mentioning that by this gestational age, the embryo has started curving into the fetal position. We've kept the ectoderm flat to keep our schematic version really simple, but these pictures from Gray's Anatomy accurately show the embryo at around 21 and 31 days. The neural tube curves through the back of them like so.
Oh, and the yolk sac and everything. Do humans... I don't know. I don't want to get into development because that'll just take too long. A similar epigenetic effect due to changes in methylation occurs in humans as well. During the end of World War II, Dutch railway workers went on strike to prevent the Nazis from bringing in more troops. Blocked deliveries of food. Okay. Hunger, winter. Over time, doctors found that the offspring of women in early pregnancy at that time experienced adverse health effects as adults. Higher obesity, higher triglyceride and cholesterol level, type 2 diabetes, and schizophrenia. Furthermore, the affected individuals had a 10% higher mortality rate after the age of 68 than their siblings who were in utero when food was readily available. Concluded that differences between the siblings' DNA methylation of certain genes caused these long-term adverse medical conditions. Epigenetic variations might help explain cases where one identical twin acquires a genetically based disease, such as schizophrenia, but the other does not. Despite their identical genomes, Alterations in normal patterns of DNA methylation are seen in some cancers where they're associated with inappropriate gene expression. Evidently, enzymes that modify chromatin structure are integral parts of the eukaryotic cell's machinery for regulating transcription. Honestly, that was not the best section. Like that didn't. Okay, if the DNA is all packed up and looped around histones, then then the whole thing about the RNA polymerase just binds to it and starts going. Well, it's gonna hit a histone. Why didn't they talk about that? Regulation of transcription initiation. Chromatin modifying enzymes provide initial control of gene expression by making a region of DNA either more or less able to bind the transcription machinery. Once the chromatin, and then also, how do you access and know where the, oh, anyways. Let's review the structure of a eukaryotic gene. Organization of a typical eukaryotic gene and its transcript. A eukaryotic gene and the DNA elements, segments that control it, are typically organized as shown in figure 18.9. Okay, yeah, so we have like distal control elements, proximal control elements, promoter. There's no operator in this thing, I guess, but whatever. They're doing the simple example. The RNA processing includes enzymatic addition of a 5' and a poly-A, as well as splicing out of introns. Yeah, associated with most eukaryotic genes are multiple control elements. Segments of non-coding DNA that serve as binding sites for the proteins called transcription factors, which bind to the control elements and regulate transcription. Control elements on the DNA and the transcription factors that bind them are critical to the precise regulation of gene expression seen in different cell types. The roles of general and specific transcription factors. There are two types of transcription factors. General transcription factors act at the promoter of all genes, while some genes require specific TFs that bind to control elements that may be close to or further away from the promoter. General transcription factors at the promoter. To initiate transcription, eukaryotic RNA polymerase II requires the assistance of transcription factors. Some TFs are essential for the transcription of all protein-coding genes. Therefore, they are often called general TFs. A few general TFs bind to a DNA sequence, such as a TATA box in most promoters, but many bind to proteins, including other transcription factors as well as RNA polymerase II. Protein-protein interactions are crucial to the initiation of eukaryotic transcription. Only when the complete initiation complex is assembled can the polymerase begin to move along the DNA and transcribe. Some genes are expressed all the time, but others are not. Instead, they're regulated. For these genes, the interaction of general transcription factors and RNA polymerase II with a promoter usually leads to a low rate of initiation and production of a few RNA transcription genes. What the fuck are they talking about? Oh, I see. Okay. Usually leads to a low rate of initiation and production of a few RNA transcription genes that are not expressed at significant levels all the time or in cells. In eukaryotes, high levels of transcription of these particular genes at the appropriate time and place depend on the interaction of control elements with another set of proteins, which can be thought of as specific TFs. So is this like a separate idea to operators? Is this like different? Enhancers and specific TFs. As you can see, no, wait. Some control elements named proximal control elements are close to the promoter. The more distant distal control elements, groupings of which are called enhancers, may be thousands of nucleotides upstream or downstream of a gene, or even within an intron. A given gene may have multiple enhancers, each active at a different time. Cell type or location in the organism. A given gene may have multiple enhancers, each active at a different time, cell type, or location in the organism. Each enhancer, however, is generally associated with only that gene and no other. Hugely qualitative statements. That's crazy. In eukaryotes, the rate of gene expression can be strongly increased or decreased by the binding of specific TFs, either activators or repressors, to the control elements of enhancers. Hundreds of transcription activators have been discovered in eukaryotes. The structure of one is shown. Researchers have identified two types of structural domains that are commonly found in a large number of transcription activators. A DNA binding domain, a part of the protein's three-dimensional structure that binds to DNA, and one or more activation domains. Activation domains bind other regulatory proteins or components of the transcription machinery, facilitating a series of protein-protein interactions that result in enhanced transcription of a given gene. Bro, this is getting confusing. Bro, this is getting confusing. Oh, here we go. Okay, this is a good picture. All right, I see. Figure 11 shows the currently accepted model for how binding of activators to an enhancer located far from the promoter influences transcription. Protein-mediated bending of the DNA brings the bound activators into contact with a group of mediator proteins, which in turn interact with general transcription factors at the promoter. These protein-protein interactions help assemble and position the initiation complex on the promoter. One of the studies supporting this model shows that proteins regulating one of those mouse globin genes can contact both the gene's promoter and an enhancer located about 50,000 nucleotides upstream. Okay, that's insane. That's crazy. What the hell? 50 kilobases away, there's this random thing that attaches? That's wild. Protein interactions allow these two DNA regions to come together at a very specific fashion, in spite of many nucleotide pairs between them. Specific TFs that function as repressors can inhibit gene. Hold on, let me look at this diagram. So we have these activators on the enhancers. I mean, it's like so fantastical, it's like hard to believe. This thing bends it in place. Wait, what just happened? Where did I just go? Let's see if we can just do some of this. Acetylation of histone tails and chromatin allows access to DNA. I think that's true. Acetylation of histone is a reversible process. I think that's also true. Some forms of chromatin modification can be passed on. That's true. Methylation of histone tails and chromatin can provide condensation. I think that's true too. Deacetylation of histone tails and chromatin loosens the association between nucleosomes and DNA. I don't think that's true. DNA is not transcribed when chromatin is. I feel like that's also true. Eukaryotic DNA is practiced. Most of the proteins in chromatin are histones. The attraction between DNA and histones is probably due to the charge of DNA. Basic structure. I think they both can be reversed. They both alter chromatin structure. That one I don't know. Neutralizes positive charge on lysine. I would say probably a seal, because methyl is just, look at this. Okay, yeah. Because this is acidic, so this is going to have a negative charge on it. Because it's given up its proton, I think. No, I don't know. I don't know what the deal is, but that was just a guess. Alright, so I think it's all of these. Fuck it, let me out. Let me out. The diagram below shows two stretches of DNA in the genome of an imaginary eukaryotic cell. Oh wait, is that true? The diagram below shows a segment of DNA containing an imaginary gene Z and the primary RNA transcript that results from the transcription of Z. Exons are represented in green. Which of the following choices represent mRNA molecules that could be produced from the primary? Oh, it might be D. Oh, it's C and D. Which of the following regulatory elements not composed of DNA sequences? Regulatory and basal. Mm. Okay, we need to watch the animation.
For a cell to develop and function properly, it must turn on the right genes at the right time. Most of the genes in a eukaryote are located in the nucleus, within a tangle of material called chromatin. Researchers are beginning to discover that the structure of chromatin dictates whether or not a gene can be transcribed. Most of the chromatin in a cell's nucleus is highly ordered and tightly coiled. In chromatin, DNA is wrapped around several types of proteins called histones. Each of the many coiled units is a nucleosome. The histone proteins have an overall positive charge, and the DNA has a negative charge. These opposite charges attract, forming a tight association between the protein and the DNA. In this chromatin structure, the DNA of the gene is virtually inaccessible, and RNA polymerase cannot transcribe it. If the DNA is inaccessible, how does a cell turn on a gene? There are actually many different triggers for initiating transcription. As one example, a cell may receive a signal during development that results in the activation or production of a specific type of protein called a regulatory transcription factor. The regulatory transcription factor recognizes and binds to exposed sequences in the chromatin of a specific gene. It may then work in conjunction with a set of proteins that can change the chromatin structure. For example, chromatin remodeling may occur through the function of a type of protein called a chromatin remodeling complex. The action of this complex is not well understood, but the resulting structural changes expose regulatory sequences in the DNA. The regulatory sequences may be close or thousands of nucleotides away from the transcription region of the gene. For simplicity, we'll examine the naked DNA of the gene. About 30 nucleotides upstream of the transcription region is the promoter, the core regulatory sequence of the gene. Next to the promoter lies the promoter proximal element. Promoter proximal elements stimulate transcription through the binding of tissue-specific regulatory transcription factors. Enhancers are regulatory sequences that may lie thousands of nucleotides from the transcription start site. Tissue-specific regulatory transcription factors bind to enhancers to stimulate transcription. When regulatory transcription factors are bound to DNA, they interact with other transcription factors. These basal transcription factors bind to promoters. They are required for transcription to occur, but they are not cell-specific and do not provide much in the way of regulation. When all of the basal transcription factors are bound, these factors regulatory transcription factors bind to enhance site regulatory sequences that may lie thousands of nucleotides from the transcription start site. Tissue-specific regulatory transcription factors bind to enhancers to stimulate transcription. When regulatory transcription factors are bound to DNA, they interact with other transcription factors. These basal transcription factors bind to promoters. They are required for transcription to occur, but they are not cell-specific and do not provide much in the way of regulation. When all of the basal transcription factors are bound, they form a basal transcription complex. One of the basal transcription factors, called a TATA binding protein, TBP, binds to a specific sequence known as a TATA box within the promoter. The TATA binding protein allows RNA polymerase to contact DNA. The basal transcription complex then recruits RNA polymerase and transcription begins. In cell types lacking the tissue-specific regulatory transcription factors, this particular gene would not be transcribed.
All organisms, whether prokaryotes or eukaryotes, must regulate or control which genes are expressed at any given time.
Regulation is essential for cell specialization in multicellular organisms, which are made up of different types of cells. Here are the key stages in the expression of a protein-coding gene and the many possible ways a eukaryotic cell can control gene expression. Let's look at the different control components and the roles that each one plays. A nucleosome is the basic unit of chromatin packing in a eukaryotic cell. One way nucleosomes control gene expression is through the unpacking and packing of the DNA, which causes a region of DNA to become either more or less accessible to the transcription machinery. The initiation of transcription is the most important stage for regulating gene expression. Transcription factors, some of which are activators, turn genes on by binding to specific
They never draw it like this. So it's always like, oh, the DNA is just floating around in the nucleus. But that's not what's happening. Yeah, let's just get through this, man. I am so done with this. The nuclear membrane's role in the regulation of gene expression involves Oh, is it this? Really? Alright, we should watch this then.
Specific regions of DNA and stimulating gene transcription. RNA processing in the nucleus also provides opportunities for regulating gene expression. The addition of nucleotides to the ends of the RNA with a 5' cap and 3' tail are important for ribosome binding and translation. Alternative RNA splicing, where spliceosome components splice out introns and join together the remaining exons, enables different proteins to be produced from the same mRNA transcript in different cells. Transporting the mRNA transcript across the nuclear membrane to the cytoplasm is an active process that requires the transcript to be recognized by receptors lining pores in the nuclear membrane.
Wow. That's pretty
Even
amazing.
after a eukaryotic mRNA is fully processed and transported to the cytoplasm, there are several additional opportunities for regulation. The process of translating the mRNA into a polypeptide also offers opportunities for regulation. Among the molecules involved in translation are a great many proteins that control the start of polypeptide synthesis. RNA-binding regulatory proteins play a key role in this process. Molecules of mRNA do not remain intact forever. RNA-degrading enzymes in the cytoplasm eventually break them down, and the timing of this event is an important factor regulating the amounts of various proteins that are produced in the cell. After translation is complete, some polypeptides require alterations before they become functional. Protein phosphorylating enzyme plays an important role in the cleavage, chemical modification, and transport of proteins after translation. The final control mechanism after translation is the selective breakdown of proteins. The length of time each protein functions in the cell is strictly regulated by means of selective degradation.
To mark a particular protein for destruction, the cell commonly attaches molecules of a
Why
small
is that
protein
so
called
loud?
ubiquitin
Stimulating gene transcription.
to the protein. Giant protein complexes, called proteasomes, then recognize the ubiquitin-tagged proteins and degrade them. This regulation allows a cell to adjust the kinds and amounts of its proteins in response to changes in its environment.
Oh,
It also
one
enables
of
the cell to
these.
maintain its proteins in prime working order.
These are actually fun, but I don't really want to do it right now. All right, let's just do it. The promoter of a gene includes the DNA immediately upstream of the transcription start site. But expression of the gene can also be affected by control elements. These can be thousands of base pairs upstream of the promoter grouped in an enhancer. Because the distance and spacing of these control elements make them difficult to identify, scientists begin by deleting sections of DNA that contain possible control elements and measuring the effect on gene expression. In this exercise, you will analyze data obtained from DNA deletion experiments that tested possible control elements for the human gene MPGES1. This gene codes for an enzyme that synthesizes a type of prostaglandin, a chemical made during inflammation. Wait, prostaglandin? Okay. There's other prostaglandin. I thought that was like a... No, I'm thinking of something else. I'm thinking of like gonadotropin or something. I don't know what I'm thinking of. Okay, the researchers about the... Wait, hold on, I have to do something. Okay. The researchers hypothesized there were 3 possible control elements in an enhancer region 8 to 9 kilobases upstream of the gene. Control elements regulate whatever gene is in the appropriate downstream location. To test the activity of one of the possible elements, researchers synthesized 4 types of DNA molecules. One DNA construct had an intact enhancer region. One construct had a possible control element. 1 deleted, 2 deleted, 3 deleted. In all constructs, the enhancer region was located upstream of a reporter gene, a gene that would be expressed at the same time as MPGS, whose mRNA product could be easily measured experimentally. The researchers then introduced each DNA construct into a separate human cell culture where the cells took up the artificial DNA molecules. After 48 hours, the amount of reporter gene mRNA made by the cells was measured. Comparing the amounts allowed the researchers to determine if any of the deletions had an effect on the expression of the reporter gene, mimicking the effect that deletions would have on the MPGS gene. The MPGS1 mRNA itself couldn't be used to measure expression level because the cells in culture have their own... Oh, I see. Wait, actually, I don't get the experimental setup. Bro, what? Yeah, great diagram. The diagrams show that intact DNA sequence... Oh, yeah, wait. This was weird. Very unexpected results. So these are actually like repressors, not enhancers or something. Did deletion of any of the possible control elements cause a reduction in reporter gene expression? How can you tell? To activate gene expression without the control element, activator is not able to bind the enhancer in the level of gene. Yeah, that's the right one. Did deletion of any of the possible control elements cause an increase? 2 and 3, or was it? Oh, it was 1 and 3 cause an increase. Both constructs result in over 100%. All right, this one was pretty easy. Some of them have been hard in the past, especially in 6A. If deletion of a control element causes an increase in gene expression, what must be the normal role of that? To repress gene expression. Okay. Dude, these homeworks are so freaking long, bro. It's the same stuff over and over and over again. Okay, we got 5 genes with their enhancers. Put an X above enhancer elements. Wait, what? Okay, I'll be right back. This stuff takes so long. Yellow, blue, green, black, red, and purple activator proteins. Yellow, blue, green, black, red, purple. Exist and combine it. Okay. Place an X above enhancer elements of all the genes that would have activators bound in a cell in which only gene 5 is transcribed. I don't even know what that means. Like, what is this question asking? It's asking you to infer which activator proteins are present in the cell. Dude, look at gene 5. Okay, that's enough for me. So gene 5, we're saying so we have these. Then we just put minuses on the rest of them. What genes would be transcribed in a cell in which the green, blue, and yellow? 4. Imagine that genes 1, 2, and 4 code for nerve-specific proteins. Which activators would have to be present in this cell type? Oh, it's just okay. 1, 2, and 4. So yellow, blue, green, black, and then 4. That's easy. Three and 5. Red, black, purple, blue, and 5. Orange. Or no, that's not orange. Okay, that's it. Red, black. In both of those questions, they didn't make you take a union. They were all, like, all of them were subsets of one of them. All right. God, these take so long. How do cells become differentiated? That sounds like that sounds like a definition. Coordinately controlled. Transcription activators. Every gene whose transcription is stimulated by a given steroid hormone, regardless of its chromosomal location. I think I might finish it tomorrow. It's getting cold. I'm getting tired. Did a lot of biology today, you know. I mean, half of it left. It's all just questions, but I do want to read the chapter because I feel like if I read the chapter, then like I'm good. You just basically gotta read this thing. Plus, I'm actually interested in this, so. Alright, but that's it for today.